Zhang from IBT. So my lab is mostly focused on bioinformatics and biostatistics.
My name's Autumn. I came down to Houston to do my postdoc, and then I realized bench science wasn't for me, and I left that, and now I work here at the GCC.
Natasha Kiriyenko, Rice University. My lab works on microbiology, model organisms, specifically C. elegans, and drug screens.
Okay, thank you all. So here's our first question. In the world of hyper-competitive science and everyone trying to publish novel results first, how do you incorporate rigor into the workflow? How do you manage the trade-offs of extra time required for rigor and results dissemination when the supervisor may want you to publish quickly?
I can start. I think as a PI, the worst thing that can happen to you if your paper is retracted, particularly, let's say, for junior faculty, it could be a decision that will break their whole career. Maybe for established professors, there are less consequences. So it's always nice to remind PI that the quality of data is very important. And I often have a couple people doing critical experiments just to make sure that we're right. I mean, there's no good data and bad data. The data are what they are. There may be something we don't know about the system, and it's absolutely worth time figuring things out.
Yes, I just want to add one comment. So I would highly encourage you as a trainee to bring open any concerns you have about your project, bring to discussing with your PI. So that way, your PI know, okay, this is something possibly we have to slow down and validate, and that's very important.
Any recent postdoc perspective on this?
No, I just think you need to communicate honestly with your PIs and say, hey, this might take longer than you think. I want to do it right. I don't want to go back and because at the end of the day, if it's your paper, you're actually the first author, right? So yes, it is hard on the PI's lab, but your name is going to be tied to that retraction, right? Because you're the first author. And I've seen it happen to some of my my friends at my PhD. Their paper got retracted while they were still in grad school. Like it was that quick of a retraction, and it was very damaging to her career.
And one comment, one of my former graduate students started a postdoc in another lab, and a year later they had to switch to a different lab because they couldn't reproduce data from previous postdoc. So it's important at some point to make a decision. If you cannot reproduce the data, do you have the courage to acknowledge that and discuss it with PI? And if PI keeps pushing that, no, you have to get exact same results, then it may be better for your future career to just go elsewhere.
And
One other thing that can help here is that actually you can point out that not just some journals, but some funding agencies are beginning to ask for this. And actually I know of some friends of mine who are working on redoing an analysis because they were trying to submit some data to the Chan Zuckerberg Initiative. And they submitted data, they said, where's the code? And the author of the first version of the code has moved to a different institution. And she had at least stopped all of her code in hidden directories. So they're now trying to reconstruct this code. The paper's already been accepted, but it's not published yet. And they're trying real hard to finish this off. But,
I just want to add kind of to what Autumn said and what Natasha said that depending on your career stage, that paper can be really critical. But I can say from personal experience that there is a time that I firmly believe that you are better off if you just pull your name from the paper. If you have a reason to believe that there is irreproducibility in the data, no matter how much you think you need that paper, it can be more harmful to your career down the road. And so I made a decision as a postdoc. Actually, it was post-postdoc that my name wasn't going to be on the paper, and it turns out it was the right decision. So it's a hard call, but in the end, your reputation is everything.
So how did you go about doing that, putting a name off the paper?
How do you do what?
Go about putting a name off the paper, like put your part actor was
So I didn't pull my name off the paper after it had been published. So, there's the circulating, it's going around to all the authors, and people were saying, yes, you got my final edits. And at that point, that for me personally, I said, I had an email trail, I'm going to go ahead and withdraw my name from the paper. So it was not after it was published, but before. Okay, next. When you inherit a project from a previous student or postdoc, how do you determine the rigor and reproducibility of their work? Moreover, if you in fact find that their work lacks rigor and reproducibility, how would you handle the situation?
Do you have any of these experiences in the audience? Because
Clearly
we can
we
tell
do.
stories, but give the microphone.
Does anybody want to talk about it? I don't want to embarrass anybody. It's like there could be people in the room. You want to? Okay. Let's see.
I cannot embarrass anyone because they're in Mexico, so I'm from Mexico, nobody knows them, so it's fine. It was not precisely postdoc, but I was having my PhD project, just started. I had come from cardiology to ophthalmology to make tissue engineering. So this was about having a product done in order to repair the corneal damage. And I inherited this work of a previous PhD that stated that changing the media or the composition of a media will produce a change in a transition from stem cells to a type of endothelial cell that we needed in order to make a construct going on. So what I found out is that this would work with rabbits, but it wouldn't work with humans. And such as is creating a project or a product that is coming for clinical, preclinicals, and so on for humans, we could not allow this to go on because it would include having an epic failure on whatever we were intending to have as a final product. So how did I find out? Well, after crying a lot because this was a year and a half later of trials and donations of cornea, you imagine it's not an easy thing to get, especially in Mexico and having human corneas going on, donors and a big amount of money involved. So I just went to my PI. Luckily for me, she's very open and she's very willing to find out a solution for that. I talked about her, showed her nothing is going on. This is a year and a half wasted of my life. I'm about to jump from a bridge. She told me not to, and I didn't. But the thing is, telling the truth and being honest is the best thing that you can do in this time. But also, I see a very good advantage on having a PI that is willing to really solve a problem that you're trying to get to with your studies. Then we had to go to the Dean of Medicine, who was the boss of everyone else. And he saw the results and he also supported us and first called the student, avoid suicide. Sorry for the true honest, but this happens. And basically I come back to the same situation, having the heads of whomever is working on your institution conscious about the negative impact that a failure of this magnitude would come. Saved me, and now we're having a startup on this, but in the right direction. So it's not everything lost. It was a year and a half. 5-6 years later, we're going in a great direction, having positive results actually in humans, not in bunnies. So beware of the species.
So I think she brings up a good point, which is I think the first thing we would all advise is you got to talk to the PI. And I would like to say that I hope most of them want the same thing that you want. What would you like to add, Natasha?
Don't be afraid. For example, I had a case when a postdoc and back then undergraduate students were working on a project, and undergraduate told me that they cannot reproduce postdoc's data. And then turns out that then nobody could have reproduced that postdoc's data. So even if the data are from somebody who's more senior than you, don't be afraid.
Any other thoughts on this? Any other experiences anybody wants to share? so this is a show of hands. Everybody get ready to raise your hands. At what point are key resources expected to be authenticated? Before publication, after data is acquired from 3 or more reproducible experiments, once the experiment does not reproduce results observed in previous experiments, or once the resource is acquired. So raise your hands if you agree with A. That's a few of you. Okay, B. Okay, C. No takers, we got one taker for C? You like all of them, you like all of the above, huh? Once the resource is acquired. So that's the one where we see the most hands. Okay, panel?
That's absolutely once, if you get something in singular of new cell line, sequence it. But if you get, let's say, transposon mutant library of 10,000 entities, then if you're going to use only 5 of them, sequence those 5. So there is a reason, but before you know your experimental outcomes, you have to know what resource was used. So when we use bacterial transposon mutants, I'll tell students that until these are sequenced or verified in some other ways that are typically validated, you don't have data because you don't know what they could be. And often that's end up true because any large scale resources will have an error rate.
So yeah, that's also NIH's guidelines. So when you write a proposal, you have to write the authentication for your biological agents. Basically, all the cell lines, all the antibody, you have to describe how you're going to validate. All the conditions. So yeah, at the beginning of the experiments.
So another show of hands here. How many of you work with some sort of cell line? Hands up high. Okay. How many of you know where there is a cell validation lab that you have access to through the GCC? So fewer of you, 3. So we'll send out the link for this, but we do have one group that does this very well at MD Anderson. For those of you that don't know, there is an MOU across the GCC, across all 8 GCC institutions, and it doesn't matter where you're at, you can access that core as if it was at your institution at the internal rates. So everybody should be thinking about validating your cells very quickly. What about, how many of you work with small molecules of some sort? Small chemical molecules, you order them from Sigma, you have them synthesized, lots of people. How many of you know where to get those validated in our area? So I see nobody. I mean, many of you that are in chemistry labs, I'm hoping that you have somebody that's at least running some mass spec or NMR or something. But we actually have a lab at Baylor that is one of the core labs, that this is what they do. They'll help validate. And I can tell you also from personal experience, when you are doing any kind of drug screens and you've ordered a bunch of analogs from Sigma and you get super exciting data and then all of a sudden you reorder it and you don't get that anymore, they don't have to provide the same level of QC for their rare chemicals that they do for some of their others. So these are things to keep in mind because it really sucks whenever you get down the road and you can't get different data. Really big fan of getting small molecules, whether you're having them synthesized or you're ordering them from a reputable vendor. You want to get those looked at as well. What else is something else you can think of that we would like to kind of validate?
Any mutations? If you get mutation or deletion from somewhere, you need to PCR and confirm that deletion is present. I had cases when I'll get mutant organisms and they would be heterozygous rather than homozygous mutants.
And I think almost all the institutions have pretty good, genetics. People didn't run that. What were you going to say?
I was going to say, this is relatively simple, but your pipettes, like if you're doing very sensitive work, then if you mispipette something, like a different amount across different drugs, you could end up with a vastly different result.
It's super true. It's been a long time since I was a grad student, but I do remember that the pipette people would come ever so often and calibrate our pipettes. Do y'all all do that? That's still a thing. Good. I don't think I must have blocked that out. But anyway.
Codes from GitHub repos.
What?
Codes from GitHub repos, or like basically codes that you get from other people.
So these are great things to be thinking about, and in the end, you can save yourself a whole lot of time. Imagine a scenario where a research study is published showing that a certain drug is highly effective in treating a particular disease. As a result, medical practitioners change their treatment approach, resulting in an increase in the number of patients being prescribed the drug. However, over time, it becomes evidence that the study's findings cannot be reproduced, and in fact, the drug may even be harmful to patients. What measures can the scientific community take to ensure that research results are reproducible and reliable, thus preventing large-scale changes in medical practice based on flawed or unreliable data?
I think prior presentation by Dr. Daggerly is an outstanding example of that. Much scrutiny is needed for clinical trials, and those of you who work with patient samples heard of IRB, so there are IDA, the FDA has separate sets of approvals, and they have really extensive guidelines. So just complying with NIH and FDA regulations would be a lot, but unfortunately, not all of the PIs follow those guidelines, and unfortunately, some of them are guidelines and best practices rather than like federal laws. So it's important, even if there are guidelines in place, to make sure that the research is in accordance to them.
You're just gonna say something.
Another issue here is that typically practitioners shouldn't be changing their practice guidelines on the basis of a published paper. This is actually where the FDA will step in, because the FDA has distinct guidelines for approval of drugs, and those have to be confirmed. And that level of analysis and checking is substantially more rigorous than journal publication. It's saying, for example, with patient trials, have you been conducting these according to a pre-specified protocol? Have you got an agreed definition of what success is, and so on like that? This issue of changing it on the basis of the paper, Lee Ellis mentioned Virgil Zola. That was on autologous stem cell transplantation for breast cancer. I don't know if any of you know of this. This was something where, for metastatic breast cancer, this guy from South Africa, Virgil Zola, said, I got great results. We should be treating people this way. And he announced this at ASCO. Some people did start changing results or changing practice that way, and actually a woman sued her insurance, which wouldn't reimburse her for it. Her family collected posthumously for $89 million, but only after they found out that they couldn't get it to work, ASCO sent out a panel to investigate his results, and he couldn't be found. And this wasn't even a published paper. This was a pre-published, but it's something we should be looking for. Has the FDA approved?
Actually, just reminds me, just quickly, a case where lots of maybe 15 years ago, saw a paper saying that cancer is not real, and then just fungal, just infection by Candida albicans. That's why it can be treated with like chalk, because chalk kills Candida. And I think these patients that were diagnosed with cancer, the provider gave them chalk, and they were fine. So just don't, be somewhat skeptical of published papers.
Yeah, I just want to comment from the other angle. Okay, so even you didn't change your protocol, and you try to increase your patients' numbers and redo the research again, redo the study, and you will find the results are different from last time. That's very common. That's what happened between phase 2 and phase 3 clinical trial. Okay, phase 2, you test a small number of patients, then show the efficacy, then you increase number maybe 10 times. Do a phase 3, then all the efficacy disappeared. That's possibly happened to 90% of the drugs go through the phase 2. So many times you don't consider this a really good issue, and that's just a validation. So that's why FDA requires you have to go through phase 2, phase 3.
So because we're going down the clinical trials path, I would be remiss if I didn't bring this up. How many of you were at our IDD conference last week? Anybody? Okay, so I hope you remember the panel that I led. I'm also going to use this as a shameless plug for another one of our our programs. But part of this is systemic. It's, you know, when you get past, you know, phase 3 and you've gone through the FDA hurdles and you've got a drug that's approved and you start seeing problems, you start questioning what went wrong. And one of the things that's really come to the forefront in recent years is that we're not getting the right data for all of the people that are going to take the drug. And so those of you that were there for my session last week and those that have participated in our diversity, disparities, and community engagement in healthcare research program have heard a little bit about some of the problems that we've seen with drugs that are approved, but you have whole ethnic groups or races that weren't even represented in the data that was collected. And it's a huge problem, one that I hope that we are really beginning to address. As much as we want to see a culture change in rigor and reproducibility, I would assert that this is a huge part of it. I mean, we do this in all of our preclinical experiments and making sure that we are controlling for everything, right? And so on that note, we do have a program in this and would love to invite you to stay tuned to watch for our fall workshop, which the date is TBD. We had launched this last year in collaboration with our community engagement folks at all of the hospitals, as well as the 2 community engagement cores at our 2 minority serving institutions, University of Houston and Texas Southern University. Super important and something that I think if you're going to be in healthcare research has to be at the forefront of your mind as we move forward. Anybody else have anything you want to say about this one? If I'm trying a protocol directly from a paper I read and it doesn't yield the same results, despite repeated efforts, do I have a responsibility to report this to the publisher or writer of the paper?
Something could be very nuanced. For example, you buy cell culture media from Sigma, and Sigma will say whether it has certain amino acids or don't, and Fisher wouldn't specify it. So you may think you're buying same media with the same name, but it will be different in composition. So emailing authors of the paper is not a problem, especially if you say, I have problems getting these experiments to work, this is what we're doing, can you please show us what could be wrong? And in this case, if there is some honest mistake, that would be helpful. And if not, at least you'll have more data then to bring up with the journal editor if this is something critical and may lead to problems for the scientific community.
Yeah, I think if I was in this situation, I would actually be really impressed that I even try to do a method from the methods of the paper. I would actually probably reach out to the authors and ask for their protocol, because there are so many things in protocols that don't make it necessarily into the methods section that maybe will change the way the results come out. So I think I would just actually reach out to the authors probably after the first time it didn't work out and say, hey, I'm trying to replicate this. I was using your methods. Do you have a protocol that you'd be willing to share with me?
And there's nothing bad about it. Recently there was emailed about staining that we used Herbst and Propidium iodide combination, that we had a specific methods article and joke, so we can also have video to highlight key points, but people still had problems reproducing, so we send them very detailed protocol. We send them the modules, statistical modules for the software, because they're using exactly the same software, and it worked for them.
Yeah, completely agree. I think that even you won't first contact the editor, possibly the first question they ask is, did you contact the authors already? So you always contact the authors first.
So another show of hands. How many of you, when doing experiments, have emailed the author of the paper and asked for help with a protocol? I know I have. Yeah. Has anybody ever had one say, I'm not willing to help you? She has, okay, yeah.
Does ghosting count?
How many of you been ghosted? Yeah. So I'll tell you a quick thing that happened to me as a grad student. You know, when you're a grad student, you assume you're doing things wrong, right? And I reached out. We had actually asked for them to share a sample with us as well as their protocol, and I spent a whole year trying to do it, and finally it's like, all right, I'm gonna do the constructs myself. And about 2 years later, the postdoc and was a postdoc at that time I had been reaching out to, he called me in my lab. He said, I'm no longer with that lab. And he said, I am so sorry because my PI would not let me share the actual construct with you that we published in the paper, and that's what I gave you the protocol for. And then less than a year later, this was like one of the first big retractions that I'd ever heard of in my career. They had to retract their structure. It was a completely messed up lab. So there are worse things than them saying they're helping you and not giving you wrong information.
I just want to add, and I think I have this right, but isn't that what the new NIH requirements are, basically obligating people to post or share their data and share their reagents as requested, or make it available, and so you have to put that into that data sharing plan.
They're supposed to have a plan for it, yes.
It's also publication requirement. Usually anything published must be shared because this is the rules of paper acceptance. But in some fields, the competitive, like let's say mouse mutations, back before CRISPR took years to make it, and then they would say, oh, all of our frozen oocytes went bad, and we don't have these mice anymore. So people in some fields used to be more protective than in other fields, but it's getting better, and it's never a bad thing to try.
Does NIH have any repercussions if you put something in your data sharing plan saying that you will share it and then you will provide it? Like, is there any recourse? Or even with journals?
I know that when people refuse to share certain strains, that editors were contacted about them, and then they would follow up to the lab. With NIH, unless using the Freedom of Information Act, you will request that PI's grant, and you'll read their data sharing plan, and you'll see that they said they'll share that reagent. Unless you do all of that, you have no leverage. But if you see that, then you can maybe contact program officer and say that they are violating the contractual obligations, because our grants are contractual obligations. So you can just then you can contact Office of Research Integrity at the institutions. There are special offices at each institution who handle grants, and that's not compliance with the grant contract. So they then will be able to reach out to PI, but that will take months.
Wow, I actually never thought about getting a copy of a grant through Freedom of Information Act. I'm like, huh.
One of my grants was requested.
You can't get the full grant.
I was gonna say, I've never did the, you know, I was
You
about
cannot.
proprietary stuff. You might be able to get the data sharing agreement, I don't know.
Well, actually, now the data sharing should be inherent. It's,
Well, you would think that the data sharing agreement should be public.
that's, and I don't know.
I don't either. Okay, so here's our next one. Is having another member of your lab repeating your experiment and comparing the results, is that a reasonable approach for reproducibility purpose?
It's better than nothing.
I agree with that. What else do we think?
It's reasonable. You cannot do two people doing everything together because that's just too many, twice as many reagents, twice as much time. But from time to time, certain experiments being repeated is not a bad idea. I had that done more in the past, especially when I know that different people have different quality of hands. Then it's nice to have somebody who does something completely different, try experiment, and then you have peace of mind.
Yeah, just out of curiosity, how many of you within your labs have some sort of protocol for some sort of second validation of your experiments? So not many, a few of you. And is it within your lab, is it internal to your lab? So you have a couple of people do the same experiment separately?
Yes.
How about somebody else that raised their hand? Is that kind of the same thing? Yeah, same thing? Okay.
But not all the experiments, of course, just the critical ones.
Yeah.
So you would fit just the main point of the story.
So you probably all know that rigor and reproducibility is a scorable component of your grants. And at one point, there was one NIH institute that was very strongly recommending a second site validation. Why would that become, for all of your experiments, why would that be an issue? Sounds great, right? Yeah, if I have a second site validate my results. Can anybody think of any reasons that that's difficult?
Twice the money.
The money, right? I mean, how many of you do animal experiments? Have you actually looked at the budgets for those? I mean, those can be really, really expensive, and if you're proposing a second site validation of everything, it basically doubles your experiment budget. And so that has been a huge issue. For a lot of people, you know, if you're working on some kind of a disease that's popular, there are several NIH programs that will test your compounds if you're doing some kind of drug research in different assays, and that can be a good second validation. Any other ways that we're doing second validation? So I would.
Sorry.
Yeah.
Just as part of the analysis thing, have two people run the code and gotten the same results before it's submitted for publication? So two people know where the data is and can run it, that's all.
Yeah. Anything else from the panel?
Actually, we do, on some assays, we do data analysis independently, but usually that happens if I look at the data and saying, no, sorry, but the statistic doesn't make sense to me. Please go to somebody else in the lab, give them the data and ask them to do it. And then usually it turns out students copy-pasted something wrong, or had a header audited, or number of animals were counted as a line. So that's easy to do, and that doesn't require extra money, and that's reasonably efficient.
That's the data for sure. I've heard that updated software or package versions impede reproducing older work of even a few years, and even when effort was made to document and preserve an analysis. How often does this occur, and what should be done to future-proof our code?
I honestly haven't had this be an issue with, I haven't had that be an issue with robust results. If you're talking about something which is near the margins, like you fought hard, by God, it's a p-value of 0.499, then you may be in trouble. But
It's very robust.
basically, the main thing for future-proofing your code, not only has somebody else run it, but has somebody else read it, because if there is the most common thing that we've seen is that some new version of the code causes something to break, just not run at all. If we have a clear understanding of what the goal was of the code in the first place, modifying is typically pretty quick. And actually, at MD Anderson, most analyses for clinical studies are run by a faculty member, stat analyst team. The stat analyst does the lion's share of the work. But what would happen is before it went off to the PI, it had to be signed off by a faculty member who had skimmed it at least, and for example, understood what the code, and I know that most of the time when I was doing this, the thing that I would send it back to the analyst for was, I don't understand your code. This needs words in addition to code saying, what are we trying to do and why?
I don't know, I mean, these were questions that were submitted by you, but if the person who submitted this question would like to elaborate, I don't know who you are. Oh, okay. Here, let me bring you the microphone so everybody can hear, because I suspect you know of a specific.
So I'll say I don't know like specific cases the same way that you're giving specific examples. I used to work with mass spec proteomics, trying to follow published results that said they did something really cool, but I was coming up with they did not do something really cool. And part of that was you have a few different software packages that had been different versions, different formatting, just all kinds of little nuances. And some of that is like little details of, okay, you updated different version of whichever program language you're running, and now you have to go through and reformat something little. Sometimes it's just that level. But I also, I don't remember, and I wish I had looked it up. I had heard a study that it might have been the similar one that you guys were referencing, that they went back and tried to reproduce results from papers that had published their data sets and said, we did this analysis, and then the second study was not able to do that, having had some of those experiences of, I know what the paper says they did, but that doesn't get, there's clearly something going on from the data, and this was because with mass spec, some of the major journals are requiring that you make available your original files. So I have the original files, and I have the paper telling me we did this software with these settings, this software with these settings, and I have no idea how they got to their published papers.
And so the excuse they're giving is, oh, it's a new version?
I don't know.
Okay.
That was one where, so the previous lab and the professors got to a point where it was, we have no idea that they're doing anything useful, their data is garbage, and that was just their, like, the lab's, my previous lab's conclusion about this other group was that they just have garbage data.
It's probably not the updated software.
So based on what you said, I can say two problems. So one is they indeed update their version, and the old version works, but the new version doesn't work, and they increase functionality. And so the problem is documentation. So they should have nicely documented all the versions so you can easily find the old version. And nowadays it's very easy, right? Suppose you use GitHub, you have all the versions saved in GitHub, so it's very easy to trace by. And another issue I can expect is, we are human, right? We always have bugs in code, okay? Almost every code you have bug. So yeah, whenever you update, possibly the new version has bug, the old version has bug, so you always have something. So that's common, okay? If that's bug issue, I mean, still, yeah, if we have both versions available, we can compare and check and see, okay, what's caused the problem? Yeah, that's my take on it.
Okay, well, next we have another show of hands question. This one makes it to every workshop because it's super important. Take a look at this and then I'll ask for a show of hands. Okay, ready? How many say A? B? Okay, a few for B. Oh, and just as a hint, this one is not all of the above. So, okay, so we got a few for B. C. D. Get off the panel. E. Okay, everybody likes E for the most part. Anybody want to weigh in on this one on the panel? I think the majority like to E. We had a few for B, and we do hear about these outlier analyses that.
So yeah, I think in the field, right, it's very common to do some statistical tests to decide if the data is outlier. So many common way to do it, 3 standard deviation, for example. We normally don't recommend, even from biostatistics field, because so the outlier possibly is caused by some biomedical reason, and you don't want to exclude the really meaningful data. So if you exclude all the outliers, so how can you make those great discoveries like penicillin, right? So yeah, so that's why the current standard is you have to document everything that possibly reasonably cause outlier, and you see, okay, that's all based on your documents, okay, can determine that outlier, then you can safely remove, and it doesn't based on any statistical way to do it.
So I will tell you that as a grad student, I had tons of data that always had this one excluded outlier. I mean, and if it can't, I was like, there's just got to be something that this outlier gets excluded in everything. And I mean, it really was an important, not published kinetics, and that I would have completely missed if we'd just thrown it out. And I was even able to look back at previous studies that had looked at similar things And their data would show this one point, you just see the curves, right? Because the program's gonna make the curves. And you could see that they were probably seeing a similar kinetic curve, but they were ignoring it because the outlier analysis said you couldn't. And it definitely made for a tidier story. I mean, it's like, oh, your data's perfect, and then these little tiny error bars, and then you got this. They were little error bars too, she said. So I personally am a big fan of E. I don't want to exclude anything unless I wrote on even, the little tiny 96 well down here, the pipette was off. I noticed that there wasn't enough in it. Whatever it is that I know prospectively I'm going to eliminate this data point. And I have a good reason.
So, I'm going to allow for a variant on E which is related to A, which is in some cases when the data do not make sense. Typically what that would do is trigger closer investigation of the data, which in some cases might identify a quite plausible reason for this. For example, working with a 96 well plate, one thing that we found was that there had been, it wasn't full, but the 4 wells on the far right side in every single plate were being overheated. Basically, the samples were cooked in those 4 wells. So it gave crap data. And it was something where we published the data both ways. We said, here's the full data, we investigated these samples, and as a result of our investigation, we decided to exclude them, and here's why.
And I've seen that in several papers where somebody says, we did exclude this for this reason, but if we included it, this is what it looks like. And I think, if you hear what Hugh says, you see this over and over again with these 4 wells. I see this over and over again with my high concentration. This is what reproducibility is about, right? I certainly don't want to ignore reproducible outliers.
But it's not always bad thing. So, for example, recently we published a relationship between colonization and host death. And we would say that if you look at it overall, in multiple genotypes, there would be no significant relationship. There would be correlation, but p-value wouldn't come to significance. But if we exclude one genetic background, then correlation becomes significant. So we said, not for all genotypes, but for some genotypes, there may be a relationship. So then you couch it in terms that there are likely different phenomena contributing, but for some of the subset, that could be valid. And I think we have both. We published both types of analysis in the paper, and then reader would be aware of our assumptions and our conclusions.
Okay, so I think this question I'm going to skip to because it's actually one topic that 3 of you submitted. I'll read the top one because I think this is basically the same. How can researchers ensure rigor and reproducibility of their methods and results in scientific studies? What steps can be taken to assess rigor and reproducibility? What are the measures we might consider if we want to make sure that our stuff is reproducible? So,
Do you know the difference between technical replicates and biological replicates? Are both included? If you're working with animals, was the power analysis done? What kind of differences you expected to detect as significant difference? Is your N reasonable? For example, with mice, usually people don't have enough animals. In C. elegans, worms are cheap. They will use 500 worms and 2% difference becomes statistically significant. So, both ways are bad. So the number of animals should be justified because you can imagine that if you're looking at 2,000 cells, 1 cell at a time, even 0.5% will likely be significant. So be very careful in the planning your experiments and making sure that analysis is robust. That's exactly why NIH and SF now requires to have statistician on a team, especially if it's something where it's quantitative or patient data analysis is involved. So careful planning and then detailed protocols.
So from the viewpoint of NIH proposal, so when we do a review, a few things we check for rigor is, do you have a presence, rigor, And how about your study design? And what about the data analysis and data management? So for your prior rigor, so basically we see, okay, do you have a sound scientific reasoning to generate a hypothesis based on your preliminary data, based on literature search? Does this alternative hypothesis exist there and shows similar possibility you didn't check, and even higher possibility you didn't check? And for experiment design, so there's lots of things to discuss and power analysis, but also many studies we found that even miss the control. So if you have, for example, you have multiple factors, did you control all the factors? And for data analysis, so we already discussed many things, but actually specifically discussed, okay, you have to include all the biological variables into your data analysis. And the data management, so we already showed, say those examples by the data management, by the labeling. So all the things related to your regular reproducibility. So that's just a principle.
And it's always important in your protocol to document criteria for exclusion of samples. Work with cell culture, we say if in DMSO control will have less than 75% cell survival after 3 days, we're excluding the samples as they too sick, and we cannot make conclusive statements about the drug effect. And it's fine, as long as it's specified and people know what exactly you're doing, then these are valid criteria for exclusion. It's not like, I don't like this sample, so I'm going to remove them.
Autumn?
I was gonna say, I think, first of all, you guys are here, so that's already a good step in the right way. But I think you just need to think critically about what you're reading, what people are telling you, what kind of data you're looking at. Like, you probably have journal clubs where people are presenting papers, like really look at what they're saying and how they did it and think like, is this something that was actually reasonable? I also think it's also very beneficial to just talk to your peers around you, people in your lab or your other grad students or postdocs that you are friends with, and say, hey, I'm thinking about doing this experiment, here's what I'm considering. And sometimes people who are even outside your field, not even doing the type of experiments you're doing, have very thoughtful observations about what your science might actually be telling you, because you're too close to the situation perhaps. And I think that just communicating with people, other scientists who can really help make it more rigorous.
We've talked about authenticating resources when as soon as you can. I'm going to put in another plug for, we have a rigor and reproducibility focused module on August the 8th on publication and reporting. Natasha and I and Stacy will be here this afternoon to teach that. And when you actually look at the things that you should be thinking about publishing, if you want to really it will guide you. And if you can do these things, it will guide you in making sure your research is reproducible, and it will also teach you how to evaluate the literature that you're looking at to determine whether or not you believe that you should spend a year trying to reproduce what this paper has said. I just real quickly before we go to lunch, anybody else, any interesting things you do to make sure that you are doing rigorous reproducible work? Any clever strategies we haven't mentioned?
Before we go, I just want to bring up very related topic. There was one question I really liked. What was the most harmful irreproducible study? And when I saw that question, it was immediately to me, Wakefield vaccine and autism study, where number of kids enrolled was too small. It was done without proper oversight, and the patient cohort was not random. They were pooled based on knowing problems with the specific children. That study was revoked, but it was directly connected to decreased rate of vaccination in Great Britain and other countries. So there are children dying now because somebody published irreproducible study. And this is number 2 most highly cited retracted paper. Number 1 most highly cited retracted paper is benefits of Mediterranean diet. So that's your connection to lunch.